and most of the time in muddy sediments. As juveniles, they live in more towards mangroves, whereas as adults, they begin to move more towards coral reefs. They are mostly noticeable by their blue giant helipeds and the fact that they are blue. This summer, I had the opportunity to have an internship in the Philippines where I studied Portunus pelagicus and their genetics for about 10 weeks. It was called the Philippines International Research Experience for Students, and I'm very, very blessed to have had that opportunity. There I learned about how important Portunus pelagicus is to the Philippines, and especially to their economy. These crabs are, in the Philippines, they are the 4th largest producer of blue swimming crab in the world, and they are actually the 2nd largest exporter of blue swimming crab to the United States. One sea called the Visayas Sea creates 40% of the exports for the country, and overall this trade creates $93 million worth of exports. So it is very important for their economy and also for fishing families and fishing villages. Because of that, it is very important to understand the genetics and population genetics so that we can manage these stocks sustainably. Obviously, we want these crabs to be used for future generations, for future economics, and just, again, environmental stability. So sustainability management is very important. Genetics can help us manage populations more sustainably because by understanding genetic makeup of different regions, you manage them differently as stocks. So in this hypothetical map, if this region has 3 different alleles of genes, and this region has 2 different alleles of genes, we want to make sure that that genetic diversity is maintained in each of those locations, which means that we're going to manage these stocks as separate stocks. So again, prevent overfishing. With that, in the past, they have researched that there are actually 2 clades of Portunus pelagicus found in the Philippines. One is Portunus pelagicus sensu stricto, which is your common type of crab, and the other is the Japanese variant. However, even though we have that CO1 research, there hasn't been microsatellite research. Save that word, we'll get to it later. However, microsatellite research has been very beneficial in areas like Malaysia, Australia, and in Thailand. In those studies, they found that there was a low level of heterozygosity, so most of the population was pretty homogeneous and pretty much very similar, and a large amount of gene flow, meaning that there was in each of those populations, there was a large mixing of genes. So our goal was, again, to examine the population genetics of blue swimming crabs across the Philippines, and it is the first study of its kind for Portunus pelagicus in the Philippines. We expected to see that there would be levels of relatedness depending on the biogeographical zonation of the Philippines. So for example, areas like the Balis region right here would be more related to areas in South Panay, but less related to areas here in Tawi-Tawi. Ultimately, we collected 186 crabs by partnering with local fishermen, and then we just extracted pieces of tissue from the chelepeds and extracted our DNA from there, and then the fishermen were able to sell or keep the rest of the crab. And we got them from 14 different locations, mainly in this Visayas Sea here, but also in Masbate, Leyte, and Tawi-Tawi, and also this Panay Sea here. To understand the population genetics, we looked at 22 different microsatellites. Big word. Microsatellites are a piece of DNA that essentially is non-coding. All of us have them, and most of the time in your genes, your gene has a phenotypic coding. So for example, like one gene may have blue eyes, or this gene might impact how like your cells work. Microsatellites have no phenotypic impact, meaning that selective pressures aren't as strong on them because there's nothing to select in natural selection. With that, every one of us has a different set of microsatellites because they are highly individualized. If we all took genetic tests right now, it would not match up because they are very much based on relatedness, and they are actually currently used for parentage tests and for criminal investigations. One other important thing to know about microsatellites is they are just repeating base pairs. So it's just like AC, 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 or TT, 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 or it can be up to 3 or 4, but they're just repetitive, repetitive, repetitive. So to ultimately study them and see what that is, because DNA is microscopic, we conducted PCR reactions on the 22 different microsatellites for all 186 crabs. What that means is that we took a forward primer, a reverse primer, which essentially help build up DNA, as well as some TAC, which is a solution that has free base pairs that can be used, DNA templates, which would be the DNA that we extracted, and then also this thing called an M13 primer tag that essentially almost flags the beginning and the end of your loci. With that, in PCR, we put all of that together and it reacts so many times to where you only have that one fragment of the locus and you have thousands upon thousands of repetitions of just that one microsatellite you want. From there, we analyzed it using fragment size analysis on an ABI 3730XL, which is essentially a needle that takes each one of those little segments and counts it using a laser. And it's really cool, and so it counts the amount of base pairs. We then analyzed these fragment size analysis using Genius. On the x-axis, there is essentially how many base pairs it has, and then this y-axis is how many total fragments were at that size. So every time you have this peak, there's essentially the majority of the fragments were at 225 base pairs long. When it is a homozygous individual, all of it is just in one peak because all of them were that same length. But when you have another allele in a heterozygous individual, because microsatellites are just adding on more repetitions of those repeats of base pairs, you just have two separate alleles that are actually different lengths. So for example, this one probably has about 10 additional repetitions of that AC, AC, AC. Having those statistics, we then looked at basic statistics and F statistics using different coding in RStudio. Ultimately, we had 177 crabs at 11 loci that were viable to be analyzed just based on heterozygosity and how clean the data turned out. And then we also looked at the number of alleles, observed heterozygosity, and expected heterozygosity. We also looked at different F statistics and chi-square analysis of those F statistics. And we found that the p-value for all of them was greater than or equal to 0.05. We'll get back to that. A little bit more on global F statistics, so we can understand the next slide. FSD refers to subpopulation to total population. This describes whether or not there is structure in the population at all. This is the main one that we wanted to look at. FIT is individuals to the total population. That is a measure of inbreeding. And then FIS is individuals to subpopulation, which is also a measure of inbreeding. So when we look back at our global F statistics, we can see that the FSD includes 0. What does 0 mean? 1 denotes that there is structure in the population, whereas 0 describes a lack of structure. So if our FST includes 0, the FIT and the FIS were also statistically insignificant from 0. When we also look back at the FSD statistics at each individual locus, we can see that all of these are also statistically insignificant from 0. And again, all of our p-values were greater than or equal to 0.5, which also just suggests statistical insignificance. So all of that being said, if 0 is over here and 1 is over here, and all of it's statistically insignificant from 0, we did not find evidence for any structure in the population for Purchunus pelagicus in the Philippines. It also suggests that the entire population is very connected around the islands. This means that this is likely due to the fact that blue swimming crabs have a very long larval stage, or fairly long larval stage of about 14 days where they're around pelagically, and so they can just swim around and have more gene flow. And then additionally, because again, 0 is a lack of structure. It does not provide any support for potential regional management. In the future, we do plan to look at mitochondrial markers. Mitochondrial markers are, again, that CO1 data that would look between the two different clades of the Japanese variant and the sensu stricto. And because of that, we would be able to see the variation in that. Additionally, all of that data has already been collected. We just have to analyze it and see if that changes the data. Also, it would be interesting to see if there were any crabs from more regions, like the north and the west, just because we looked very central Visayas area. Also, this, I just want to point out, is with the Monterey Bay Aquarium Seafood Watch, and there actually are, we correspond with them a lot to give them this information so that they can help those individual fishing villages make sustainability plans based on the research that we're doing. But yeah, I just wanted to give a huge thank you for all of you guys for coming, but also to the National Science Fund for funding this research, for FIRUS, and then also for Dr. Mamnalia, Dr. Sir Percy, and Dr. Goudier, and Dr. Bird, especially, just for selecting me for the program and for it being just a great experience. And then also for Jazz and Roxanne. Salamat kaayo. But yes, thank you guys so much. Any questions? Also, I have a reference list printed if any of you would like to see references, but yes.
What do you think was the most challenging aspect of the study?
In general, coding. It's also the proudest part of the study because I coded all of my own statistics, but yeah.
Are there phenotypic differences between the clades? I mean, any expectations?
No.
Does one grow bigger or anything like that?
No, there are no phenotypic differences between the clades, just CO1 mitochondrial differences.
Okay.
So
it seems like they would have the same selective pressures.
Yeah.
Certainly be on an island, gene flow very easy. So
yeah.
Makes sense.
Yeah.
I just was wondering, what prompted like the start of this study? Was it like to prevent them from becoming too like homogeneous, or was something happened in the past that they want to prevent from happening again? Or?
Yeah, so I don't know that there was anything specific in the past, but I do know that actually Sir Percy had started this research in the Philippines because it had been done in like Malaysia, but he mainly noticed the two different clades that showed that there was more genetic diversity than they thought. And so the microsatellites was just a follow-up of being like, okay, if they're showing mitochondrial differences, what are actually the differences in the microsatellites? But yeah.
Why was it important to study microsatellites specifically instead of other non-floating nucleic acids?
Yeah. I know that one of the, I am not aware of other non-coding genes, but I do know that part of the big part with microsatellites is that there were already microsatellite primers that were made for blue swimming crabs, and so we didn't have to make any new primers. We just tested the 22 that were in the literature, and that was also a really big part, was just the fact that we tested it on a larger sample size because all of those primers had previously just been tested on a group of 30 crabs from Malaysia. Crystal,
so from your results, you mentioned that it supported the idea that there's like a lack of population structure and there's a lot of gene flow. Are there any policies that are in place right now, environmentally speaking, that permit that to continue to have that gene flow?
Yes, so the Philippines is very good about especially doing there are several governments, I can give you a link to like some of the articles that are written in their law about just sustainability in general, and so that's why the Monterey Bay Aquarium Seafood Watch is very involved in helping that sustainability, but one thing that I really like about it is that they're very intentional about village to village, seeing what impacts it has. Yeah. Thank you very much.